Greetings, this is Dr. Jess, and I'm making this short video to answer the question, where is the evidence concerning live blood cell analysis? This is a question that is consistently asked of me, and I'd like to address it in the following way. If you look into the literature and type in live blood cell analysis or live cell analysis or any of these terms that we generally use, you're not going to find too much. But if you look at various studies, they utilize various microscopy techniques in order to gather their data for their studies. Live blood cell analysis is nothing more than a microscopy technique, and it's a microscopy technique that can be brought right to your office and done for the benefit of your patients. But since everybody wants scientific evidence, let me prove my point. In the study VMP-35MNC, a novel iron-free supplement enhancing cytoprotection against anemia in human subjects, a novel hypothesis that was published in Food and Nutritional Research in 2019. Under methods, live blood cell imaging was performed by the Veritas Health in Woodbridge, Ontario, Canada, using the Olympus BX30 light microscope with a phase contrast condenser to visualize samples. This is a peer-reviewed scientific journal, and it was recently published, but I'm highlighting the fact that live blood cell imaging was performed in order to gather the data for this study. In the same study, under study design, it states that changes in peripheral blood smears were observed during using live blood cell imaging and phase contrast microscopy. Again, our live blood cell analysis and dry blood cell analysis course is to teach you how to utilize microscopy to determine the condition of someone's blood. In the research paper entitled Observation of Dynamic Subdomains in Red Blood Cells that was published in the Journal of Biomedical Optics in 2006, it stated in the abstract that phase contrast microscopy has been used to study spatially resolved red blood cells. Please note that phase contrast microscopy is live blood cell analysis, just a different name. In the study Quantitative Phase Imaging Techniques for the Study of Cell Pathophysiology, the authors explained that quantitative phase imaging, or QPI, is one of the optical microscopic techniques, and it's being investigated and widely utilized throughout the scientific community, generally because it's easy to use, it's accurate, and we don't have to use dye or fluorescent proteins, stuff like that. I thought it was very interesting that in this research work highlighted in this article suggests that the various QPI methodologies may play a critical role in answering contemporary questions in the pathophysiology of cells and tissues, which could indeed bring a substantial improvement in the understanding, assessment, and treatment of diseases. This is exactly what we teach, but because we call it live blood cell analysis, everybody says, where's the scientific evidence? Well, it's sitting right here staring at us, people. So as you can see, we've been looking for the scientific evidence for live blood cell analysis in all the wrong places. The intrinsic worth of live blood cell analysis is so obvious that there is no need for studies of live blood cell analysis in and of itself. Scientists around the world are using this technique by other names or using different equipment, but essentially it's looking at live blood cells under the microscope to get the data for their studies, which are being published in peer-reviewed scientific journals. I think that qualifies as letting us know that live blood cell analysis is a valuable technique. And if you can bring this to your patients in your office, clients, patients, whatever you like to call them, and gather information that will improve your diagnostic acumen and improve your patient compliance and make better treatment plans and ultimately get people better, quicker, more efficiently, I'll leave you to make the appropriate conclusions of what that would do for your practice and for your own feeling of self-worth as a practitioner. So our next course is 27 and 29 September. It's in Cheltenham, UK. Okay, for more information on the book, contact Carol at itteacher.co.uk. Ask her for the flyer. If you'd like a better explanation of the course itself, there is the video that I made, which is on my YouTube channel. And I hope to see you at the course, because there is now no reason, any scientific reason, that you shouldn't be thinking about adding this to your armamentarium when it concerns the care of your patients. Take care.